I'm going to be going over DNA replication. So the first step is we have topoisomerase helps, you know, relax the supercoiled DNA. This helps unravel it a bit. And then we have helicase, which is this right here. What it's gonna do is it's gonna come at the replication fork, and it's going to bind to the DNA at the replication fork, which is this whole middle, this Y kind of shape here, and it's going to untwist and unzip the DNA, break the hydrogen bonds so that we have these single strands of DNA that are going to be copied. And before they're created, before DNA is going to be copied, we're going to have the single strand binding proteins. And what they're going to do, these purple proteins right here, what they're going to do is they're going to stabilize the DNA. They're going to make sure that the strands don't bond back together. Step 2 is elongation. You have DNA primase. It creates RNA primer, and what this does is it makes this enzyme right here, this really important enzyme, DNA polymerase. It helps signal to make sure, like, because what DNA polymerase does, and it creates pretty much the DNA. And what happens is when RNA primer is put down here, DNA polymerase is going to add a nucleotide to the RNA primer and make polynucleotides. It has two functions. The second function would then be to remove the RNA primer and replace it with DNA. And you've got these DNA copies here. DNA ligase is then going to seal the gaps in the DNA because there are going to be gaps because of the Okazaki fragment. And well, you have these thing here called leading and lagging strands. And this happens because DNA is anti-parallels. So we have one side's going to be 5 prime, 3 prime. The other side is going to be 3 prime to 5 prime. And we have the leading strand here because, it leads, and then you have the lagging strand because it lags behind, because DNA can only be built in the 5 prime to 3 prime direction. So the leading strand has no problem doing that. DNA polymerase can go straight through and create DNA perfectly, but with the lagging strand, the DNA is built the opposite way of the leading strand, and because it can't build that way, the RNA primer is going to have to keep signaling the DNA polymerase back so it builds in this direction here, and it's going to have these fragments because the DNA polymerase keeps going back, and it's going to create these fragments called the Okazaki fragments. The third step would be termination, and what happens at termination is you've got you have telomerase, and it's an enzyme that creates telomeres. And what happens is telomeres end up eroding. The ends of chromosomes are eroded with replication, and it helps protect the ends of the chromosomes. And that would be it.